Hello, my name is Sarah Dean, and this is video 5 in a tutorial series on using IGV. In the previous video, I went over how to distinguish sequencing artifacts from true variants, but I focused on single nucleotide polymorphisms and small indels. In this video, we will look at structural variants and discuss how to interpret them using IGV. Structural variants are mutations that disrupt larger chunks of DNA or the structure of a chromosome. They include gene fusions, translocations, inversions, internal tandem duplications, and very large insertions or deletions. I don't have examples of all of these types of structural variants, but I will go over how to look at all of them, and then we'll look at examples of a small subset. But first, I have to go into sequencing chemistry a bit. This is another place where it is important to understand something about the chemistry that generated the data you are viewing. Alignment interpretation may change a bit with a different chemistry, but even if you are using a different chemistry, hopefully this segment will at least demonstrate how to think about chemistry in the context of DNA alignments. Our data was generated by Illumina's NextSeq 550. These sequencers have oligonucleotides covalently bound to a flow cell in the sequencer. During DNA library preparation, complementary oligonucleotides are ligated to the ends of the DNA fragments so that when they wash over the flow cell, they bind to the oligos on the flow cell through complementary base pairing. The oligos ligated onto the DNA fragments are called Illumina adapters, since they are required for the DNA libraries to be captured on the flow cell of the Illumina sequencer. Once attached to the flow cell in this way, sequencing by synthesis begins for 150 cycles in our case. So 150 nucleotides are sequenced in one direction. Our data is also generated by paired end sequencing, which means after 150 cycles, a turnaround happens so that the opposite end of the DNA fragments can be sequenced. It is not the purpose of this tutorial to teach sequencing chemistry, but the abridged version of how this happens is that something called bridge amplification occurs, and during this process, the DNA becomes secured to the second oligonucleotide and can be denatured from the first oligonucleotide it was originally bound to. It is now upside down relative to how it was oriented before, and sequencing commences from the other end for 150 cycles in our case. This second round of sequencing creates the paired end read to the first round of sequencing. In this image, the purple represents the Illumina adapters that allowed the DNA sequence to be captured on the flow cell, and the light blue represents the DNA fragment we are sequencing. This is called the insert, as if it were inserted between the two adapters. Some inserts are longer than 300 base pairs, which means that 150 sequencing cycles in each direction is not enough to sequence the entire insert. Read 1 and read 2 will not overlap, and there will be a gap between them when they are mapped to a reference genome. This gap is called the inner distance. On the other hand, some inserts are short, less than 300 base pairs. In this case, the paired reads will overlap and there will be no inner distance. The shorter the insert, the more overlap between the paired reads. Structural variants are difficult to visualize in IGV because the subject's DNA that includes the structural variant is mapped to a reference genome that does not include the structural variant. To notice structural variants, we have to keep track of read direction, primer direction, and insert size. When these are not the expected size or orientation, it often indicates the subject's DNA is being warped in strange ways to force a fit to the reference genome in IGV. It's up to you to figure out how the subject's DNA is being warped and determine whether that means the subject's genome contains a structural variant. We know that read 1 and read 2 in paired end reads always face each other. So if read 1 and read 2 do not face each other when they are mapped back to the reference genome, it suggests that something strange is going on in the subject genome, such as the inversion that is shown here. An inversion means a segment of the subject's DNA is flipped backwards, indicated by the lettering here, which is AB in the reference genome and BA in the subject's genome. The arrows at the bottom here represent paired reads produced during sequencing of this inversion at the breakpoints. Because the segment in purple is flipped from the reference genome, when aligned to the reference genome, the arrows from the purple regions will get split up and flipped around, as indicated in the upper image. When paired reads get split and flipped like this during alignment, it is indicative of an inversion. You can tell IGV to color code paired reads that are not oriented properly so that you can notice them easily. And I will show you how to do that. Remember, all you will see in the alignment is the paired reads mapped against the reference genome. You won't see the subject genome in IGV. To figure out what is going on in the subject, you have to remember that in reality, read 1 and read 2 from paired end sequencing do face each other in the subject, as shown in the subject image at the bottom. But it doesn't look like it when mapped to a reference genome, which is not representative of this subject. This is why structural variants are difficult. We have to reverse engineer what the subject DNA looks like from the distortions that we see in the alignment. Many reads from the inverted segment in the subject can align well to the reference if IGV flips them. And the IGV will do that, especially since inversions are often very large. Their entire length is probably not spanned by a single read. Instead, it may be composed of several separate reads, indicated here by the orange lines, many of which can simply be flipped to get them to align without a big mess. So the reads lying entirely within the inversion or entirely outside of it will carry no mismatched nucleotides indicating a mutation, as we are used to seeing. But in the case of reads that span the inversion breakpoint, part of the read will align well to the reference genome and the other part of the read will not. These reads are now indicated in red. IGV will align these in the orientation that maps best to the reference genome, but the portion that does not map to the reference genome will be very colorful because none of the nucleotides will match with the reference genome in that segment of the read. The black lines here indicate the division point between full alignment to the reference and complete non-alignment to the reference. These are visually obvious in IGV, and they indicate the genomic positions of the breakpoints for the structural variant, in this case, the inversion. These poorly aligned insert ends are automatically hidden by IGV. This is because IGV automatically hides what it calls soft-clipped bases. It does this because many sequencing chemistries produce poor quality insert ends. Ignoring these error-filled ends generally creates better alignments, and that was fine when we were looking for small mutations because it removed some of the noise that obscured them. But it is not good to hide soft-clip bases when you are looking for structural variants because the soft-clip bases will show you where the breakpoints are and help you understand your structural variant better. I will demonstrate all of this in IGV after we conceptualize some of the other types of structural variants and how IGV distorts them to force alignment to the reference genome. So now let's suppose the subject has an internal tandem duplication, or ITD, that is so large it cannot be spanned by a single read. The duplication is not going to show up in IGV clearly because IGV is going to collapse it as best it can to align it against the reference genome that does not have a duplication. So again, we will have to use clues to detect an ITD. Again, the arrows in the lower image indicate the forward and reverse reads, or read 1 and read 2, from paired end sequencing in a patient with a structural variant. This time, an ITD. The image at the top indicates how these reads are oriented when forced to map against the reference genome that does not contain an ITD. Even though paired reads always face each other, when mapped to the reference, they appear to be facing away. And this is because there is only one position A in the reference and only one position B, while in the subject, positions A and B are duplicated. The image at the top indicates the expected genome, and the image at the bottom shows a subject with an inverted duplication. The black arrows represent paired reads whose position does not have to change to be mapped to the reference genome, because the subject genome is as expected here. The turquoise and purple arrows are similar to those shown in the earlier inversion. The difference is how far one of each pair is found from where it is expected to be. The reads encircled here are far from their mates, indicating that the region they have mapped to has been repeated where their distant mates are. Examination of the unaligned soft-clip bases can provide further support for a structural variant like this. Finally, here's an example of a translocation where a gene or a part of a gene has been moved to an unusual position on the same chromosome or perhaps onto a completely different chromosome. Again, you will not see the subject's genome. You will just see the paired reads appear to be unpaired when forced to map against a reference genome, which is arranged differently from the subject. The color coding of the arrows throughout this tutorial corresponds to the color coding IGV uses when you tell it to color reads by pair orientation. These images are from IGV's open source help page. The only exception is for translocations across chromosomes, called fusions. IGV has a color code for each chromosome, so when pairs are mapped to different chromosomes, they will carry the color of the chromosome of the mate. Additionally, reads can be colored by unusual insert size, which may indicate particularly large insertions or deletions. IGV calculates the average insert size across the entire dataset and the insert size standard deviation, and it uses this to identify inserts that are statistically significantly different from average. It has to infer insert size based on the reference genome because, remember, many inserts have an unsequenced gap between reads called the inner distance. When an insert is particularly large or particularly small, it may be because the unsequenced portion contains an anomaly, such as a deletion or an insertion. So putative deletions are colored in primary red and are represented by unusually large inferred inserts, while putative insertions are colored in primary blue and are represented by unusually small inferred inserts. This may seem counterintuitive, and that is because, again, we are not viewing the subject DNA, but rather how the subject's DNA is warped when forced to align to the reference genome. On the left, we are looking at a large deletion in the subject. Now, most inserts will, in reality, be approximately the expected size, just based on the laws of probability. You can see that in the bottom left panel. The two reads there are creating an insert of the expected or average size in the subject. But because this subject has a large deletion, meaning part of the genome is missing, when it's mapped back to a normal reference genome, the paired reads get stretched out from each other, and the inferred insert size appears much larger than expected. Now, very rarely, inserts will be truly larger than expected, but more often they are not significantly larger. They just look large when they have to be warped to align to the reference due to a deletion. Insertions work in the reverse and are shown on the right. If the subject carries a very large insertion, even though the insert represented by reads 1 and 2 are actually of normal length, they have to be squished together to be mapped against the reference genome, which does not carry the insertion. This gives them an inferred size that is significantly smaller than expected, which is why unusually small inserts are actually suggestive of an insertion. I know it's kind of confusing to have insertion and insert, so remember, an insertion is an added chunk of DNA into the subject's genome, while an insert represents the amplicon that we are sequencing. You might see this red and blue color coding in IGV show up when you are looking at other types of structural variants that we've discussed already. And this is because other types of structural variants can cause an insert to have an abnormal inferred length when they bring genomic positions closer together that are normally far apart, or create distance between genomic positions that are usually close together. So now that we've talked about what structural variants are and how they become distorted in IGV when mapped to the reference genome, let's look at a couple of real examples. We've already talked in previous videos about how to load files into IGV and the different file types. I just want to point out that I have 2 different BAM and BAI files for this particular sample. And that's because our secondary analysis software produces a BAM and BAI that are optimized for looking at SNPs and small mutations, and it makes another BAM and BAI for looking at structural variants. So I'm going to choose the structural variant files, and I like to have the GTF file here, which shows where the primer binding sites are. I think it just gives some context for looking at the alignments. Now, this tutorial is not intended to show you how to identify structural variants de novo. My secondary analysis software has already identified a structural variant and its breakpoints, and my goal here is just to look at the structural variant and determine whether it's real or not. So my software has indicated to me that there is a structural variant in chromosome 13, and I'm going to enter the position of one of the breakpoints here just to get to the general area. So always what I do first is go to Region Navigator. So if I go to the Regions tab and Region Navigator, I can add a line. And in these fields, I can enter the putative breakpoints for this structural variant, the start and stop points. And after entering the breakpoint, I have to click Enter. If I just click out of this field, it'll revert back to what it was auto-filled with initially. All right, and then I click View. And now there's a new bar at the top here. So if I zoom out, you can see this bar represents the putative breakpoints for the structural variant identified by our software. So now we're going to see whether that makes sense in light of the alignment. So the first thing you want to do is make sure you've color-coded your alignment by insert size and pair orientation. So we just finished talking about how insert size and pair orientation can be useful things to look at when identifying structural variants. And then I also want to view as pairs. So now read 1 and read 2 for the same insert are always together. Now another thing we need to look at is soft clip bases. And these are already showing for me, but IGV hides these automatically. So for you, you may not be seeing them. So I'm going to show you how to change IGV's view settings to always show soft clip bases. So we go up to View, we go up to Preferences, and then we select the Alignments tab here, and you just scroll down for soft clip bases. So see, for me, it's already checked, so you'll need to make sure that this is checked for you as well. And then just click Save. And once you do that, from then on out, soft clip bases will always be shown automatically, right? And the last thing I want to do to set myself up for looking at structural variants is to make sure that the center line here is right at the edge of one of the breakpoints. And then I'm going to group alignments by pair orientation, all right? So now you can see, remember, these blue are indicative of an inversion. And if you don't have a center line, in a previous video, I describe how to make the center line show up. And then I also want to sort alignments by base. So that, now I've just brought all those soft clip bases to the top. So the soft clip bases are where nothing aligns to the reference genome. And though we spent a lot of time talking about pair orientation and insert size, it's actually the soft clip bases that are your strongest evidence for a structural variant. The evidence for a structural variant that I'm seeing here is that these reads align perfectly to the reference genome up to a point, and then there's no alignment at all, very abruptly. So if these were just messy ends, first of all, you wouldn't expect them to all be the same like they are. So these all align very well with each other. They just don't align to the reference genome. So that suggests that this sequence here is real in the patient. In this case, it's not a patient, it's a synthetic control. But you could be looking at a human genome, so I'm just going to say patient. So this sequence here, shown by these soft clip bases, appears to be real in the patient because there's a lot of support for it. It just doesn't match with anything in the reference. So the reason IGV typically hides soft clip bases is because it's not uncommon for the ends of reads to be of poor quality and therefore to align poorly to the reference genome. And so IGV usually hides the soft clip bases. However, here, there's no reason to think that this is a mistake due to sequencing. It's consistent across all these different reads. The quality of the reads are high. So that's why we want to look at them, because in this particular case, they're showing real information about the patient. They're not just displaying sequencing errors. So now I'm going to show you a few tools for a deeper investigation of this putative structural variant. All right, so first I'm going to raise up this bar because I need to make space for new tracks down here. And I think it's best to select one of the longer soft clip bases, and I'm going to right click on it here. And if I go down here, I can see that there's an option to BLAT left clipped sequence. So the left clip sequence references the soft clip bases on the left side of a read, and BLAT stands for blast-like alignment tool. So basically what we're doing when we select this is blasting the soft clip base against the reference genome to see where it actually belongs, if anywhere. All right, so a little window pops up showing you some score information, a start and end. You'll notice a new track has shown up. And so I had selected this longer soft clip base here, and now it's showing me where it aligns. So it actually does align to the reference genome, but it doesn't align to this part of the reference genome. So what's actually happening in the patient DNA or in the DNA of this control is that this sequence is normal until here. And then starting where this red mismatched base appears, this part of the genome repeats itself again. So this is just a repeat of this section here. So if I zoom in, that makes it a little bit easier to double check. So again, this is the sequence that I blatted. So it starts with a TTAC. So if I go to where it says that it belongs, TTAC, right? And so if I look carefully, I can see that this misaligned portion actually aligns here. It's just this portion repeated. So this shows you that there is an internal tandem duplication. And this internal tandem duplication is very short. It happens within the span of a single read. So I can already see that the rest of these soft clip bases are the same as the one that I already blatted because they all have the same color coding. So the color coding reveals that. But we can build our confidence in that by blatting more. All right, this one looks a little bit different. So maybe we would want to give that a try and see what happens. And I'm going to need to make more space by pulling this up. I'm going to delete a couple tracks. So to delete a track, you right click on it and remove track. Because these are redundant, right? They're all telling us the same information, which is that this set of soft clip bases aligns to this read here. And then this set of soft clip bases appears to align to this set here. So this is making it look like there's two different breakpoints, and actually there are. So the first set of soft clip bases up here, they all aligned to a breakpoint here, which does not match up with the breakpoints I put in this bar in red using the region navigator. But now this next set of soft clip bases does line up with the breakpoints I entered into the region navigator. Now, this particular synthetic control does in fact have two different structural variants in the same location, and one of them is larger than the other. Now, the one I entered up here is the smaller of the two structural variants that are found in this location. Now, sometimes a structural variant will align to places on multiple different chromosomes. It's probably wise to select the match that has the highest score. All right, now obviously things start to get kind of crowded and this is all redundant information. So I'm just going to keep this one because it has the most data. It's the longest soft clip base that I've blatted so far. And so again, I can double check what's happening here by looking more directly at the sequence. So the soft clip bases begin here with a quadruple C, G, triple A, and then this is supposedly where they align to, and you can double check that. So now that I'm looking more closely, I can see that actually the breakpoint begins at this triple A, and this quadruple C here, this is just a poorly aligned region. So you can see there is a triple C here, and sometimes in all of these sequences, even those that are not part of this structural variant, this A is sometimes read as a C. So that can just be considered sequencing error. But starting at this triple A, the duplication begins. So the patient's genome is normal, 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 up until about here. And then here, this triple A is repeated and then just keeps going and going and going. So these breakpoints, they indicate the segment that's copied and duplicated again over here. But even though this segment that's copied is small, it ruins the alignment for a much longer region, right? Because everything after that is shifted over into the wrong place relative to the reference genome. All right, I almost forgot to come back to these inversions at the top here. So we can blat these as well to see what's going on. I'm going to start with, these are larger over here. All right, so it matches to chromosome 13 and chromosome 2. We're going to select 13, so stronger alignment score. All right, so let's back out a little, see what that looks like. So this reverse read over here aligns to the forward reference genome, forward strand on the reference genome over here. So that certainly seems like an inversion. So the blat soft clip base tool is only available for a certain size of soft clip bases. Under a certain size, that tool becomes unavailable. But we have other options available to us. So this is a good opportunity to look at this. So instead of blatting soft clip bases, we're going to copy the left clip sequence. And then we're going to use a new tool called Find Motif. So here's the Find Motif tool. And we're just going to paste using, hotkeys, Control V. All right. And so it automatically fills the positive and negative strand. It's going to find the reverse complement on its own. All right. So this matches to over here. And it's not showing you the arrows to indicate whether it's forward or reverse, but you can see it actually creates two new tracks, one for the positive, the forward strand, and one for the negative strand. So this is on a reverse read, but it matched to the forward strand on the reference genome. I'm going to remove the empty track here. All right, and then over here, we'll do the same. So again, blat soft clip bases or blat left clip bases, that's unavailable. So we're going to copy left clip sequence. We don't want to blat or copy the whole sequence, right? We just want the part that's mismatched to see where it belongs. All right, Find Motif tool, just copy and paste here. All right, where does that show up? I'm going to have to pull this up, I think, to see. All right, so this one is a reverse read that aligned to the reverse strand of the reference genome. I'm going to remove this empty track here, pull this back down. All right, so this does seem to be indicative of some sort of inversion, but there's not very many sequences supporting, right? So it's hard to say. There's only 2 inserts supporting an inversion. This inversion was not called by our software, probably because there's just not enough evidence for it. All right, so I'm going to clear the screen, and next we're going to look at a much larger structural variant. All right, so here I just navigated to the gene of interest by writing the name of the gene up here. You can also do that. I'm going to go straight to the region navigator here and add in the breakpoints given to me by our software. All right, so enter and view. And it automatically zooms in on the regions I've entered in the region navigator. So here's our structural variant. This is going to be a much larger structural variant than the last one. So again, I want to color code by insert size and pair orientation. And I also want to view as pairs. So you can see there's a lot more going on here, lots of different colors, more regions of soft clip bases. So now we have to think about how to organize all of this. I want to sort by pair orientation, but you have to put the center line on the genomic position that's going to be used for sorting. And that's the part that's sort of difficult with this large structural variant. I think this kind of spans all of the different pair orientations I can see. So let's see. Group. I want to group alignments by pair orientation. All right. So these darker blue reads suggest inversions. These light blue reads also suggest inversions. And these green reads suggest internal tandem duplications, but they can also suggest translocations. And there are a lot of them. And one thing you might notice right off the bat is that a lot of these soft clip bases are lining up really nicely with the breakpoints in the region navigator. So for a structural variant of this size, it's going to be very challenging figuring out exactly what the sequence of the structural variant is all the way through. You might question, is that really important to do? Are you just interested in whether this gene is still functioning properly, or are you interested in exactly what event took place? At the end of this tutorial, we are not going to know the exact sequence of the structural variant. We're just going to explore some of the things that are going on here. All right, so I'm making some space for new tracks here. And I'm going to start with the low-hanging fruit. So I'm going to start with where this is the piece that has the most support, right? So let's take one of these longer soft clip bases, and let's actually zoom in a little. So these all look well aligned with each other again, right? Based on the color coding. So I'm going to choose a soft clip base that seems fairly representative of the rest, and also kind of on the longer side, maybe see where it belongs. All right, so this tells me where that is, and it took me to it, right? Now, remember, again, this is a very large structural variant, so it took me pretty far away from where I was originally. All right, so these over here, these soft clip bases, belong over here, and they face in the same direction. Now, also notice read 1 and read 2 face away from each other. They are in the patient actually facing each other, but when aligned to the reference genome, they face away. All right, so let's do a few more just to build support. All right, there we go again. Same place. And so if I were to scroll down, you can scroll down to see if you see anything unique, but all of this pretty much looks the same. So I'd expect it to all align to the same place. I'm going to take a smaller one. All right, there we go. So we can just assume now, I think, that all of these belong over here, just inside that breakpoint. All right, and how about these soft clip bases on the right then? Let's blat the right clip sequences. Where do they show up? So they haven't flipped, you know, these look like forward sequences. They're remaining forward, but it should be over here. All right, and they'll probably all be the same too. So the right soft clip bases belong just inside the left breakpoint. The left soft clip bases belong just inside the right breakpoint. They have not flipped directions, suggesting that there is not an inversion here, but instead there's a segment of the patient's genome that is being copied where it shouldn't be. Now, in the patient, these forward reads are normal until here, and then a section of the genome all the way over here has been copied over here, right? So there's this very far away section from the same chromosome, but that got copied over here. So what's actually probably happening is, if you could imagine taking this reverse read right here, just lifting it off the page and putting it here with its mate, the part that's a soft clip base over here would turn green because it would be properly aligned if you placed it here. And then the rest of its length would become a soft clip base over here because it actually aligns over here. So this is showing you again that these reads actually in the patient, they are facing each other and they probably overlap in reality, but this piece of the reference genome has been inserted into the patient's DNA over here. So these green reads can be representative of a duplication or a translocation. And it's really hard to tell with a structural variant of this size what's going on in the middle to fill in those gaps in the data. So it could be that these can help fill in some of those gaps. But like I mentioned, we're not going to fill in all the gaps. We're not going to get the entire sequence for this huge structural variant. As already discussed, these dark blue and light blue reads are representative of inversions. To understand why, I'm going to blat some soft clip bases. And I'm just going to do one from each side to save on time. Just take, one of the longer sequences, maybe one of these. So in these dark blue paired end reads, the forward read and the reverse read are both mapping primarily to the reverse strand in the reference genome. That's why they're both facing to the left. But these soft clip bases on both the forward and the reverse reads are mapping to the forward strand of the reference genome. Now remember that in the patient, both the forward and the reverse read actually face each other. They're only facing in the same direction because of how they map best to the reference genome. So you have to imagine that in the patient, this is actually flipped around and these soft clip bases line up over here. What that means is that in the subject, these reads are facing forward as they should, but this segment here is reversed from how it's supposed to be in a normal genome. Now, the same is true for these light blue forward and reverse reads, but in reverse. So I'm going to blat two from the same read. So these soft clip bases on the forward read map to the reverse strand on the reference genome. And that second set of soft clip bases don't map. It's chromosome 19. It's not the first hit. It's actually not in the range of where we're looking right now. So these map to somewhere totally different from where we're looking. So they don't really provide a clue as to what's going on here. When that happens, it's hard to know how to interpret that. If it's not the main story, it's best to just ignore it. If we were to take the time to blat all of these, they would all support a story of something being inverted within these breakpoints. If you have support from a lot of different reads, that's one thing. But if it's just one stray read, you have to ignore it. All right, so what we have so far is information about there being a large internal tandem duplication that contains an inversion. And if we wanted to fill in the middle of these breakpoints a little bit more, we could scroll down And, these are soft clip bases that sort of line up with those that we were already blatting. And then here's some soft clip bases from inside the breakpoints. So this suggests an internal tandem duplication that's repeating. So we know that there's a region over here that's repeated over here, but these left clip bases are repeats from the reference genome over here. These soft clip bases are a repeat of this part of the reference genome, and these soft clip bases are a repeat of this part of the reference genome. Also, read 1 and read 2 are facing away from each other when they should be facing each other. Remember, that's suggestive of a duplication. So the data that we're collecting now suggests that there's multiple duplications of the reference genome between these two breakpoints, as well as an inversion. And that's probably as far as we can reasonably go with this software. Now, perhaps there are some applications where you would want to know what the exact sequence is in this large structural variant, but the practical implication is that this gene is no longer functioning as it should. And the truth is, you could figure that much out by a quick glance at how much support there is, as well as the size of the structural variant. All right, so now if I scroll all the way to the bottom, just to see, it's good to scan to see if there's anything new. Down here are some interesting color codings. So this has to do with suspected fusions. Now, I don't believe any of these fusions are real, just because there's no consistent story going on here. But these are color-coded for the chromosome the mate belongs to. So I had said earlier in this tutorial that I would show you how to look at fusions. So even though this fusion isn't real, we can still look at how we would interpret fusions here. So I've cleared out the tracks. So if we had a real fusion, they would look something like this, although there would be more consistency across inserts. Each one of these is colored for a different chromosome, which is why I don't believe any of this. So if you were to right-click on here, you can either go to mate or you can view mate region and split screen. So when you're looking at real fusions, this is very useful because all the mates should come from the same chromosome. So you would just split the screen like this and have the mate show up. But see, the problem is that I'm all the way at the bottom of my alignment here, and the mate is going to show up all the way up here. So here's the mate highlighted for me. So that's in chromosome 2. So we have a couple that are blue here. We can look at that as well. I've split the screen again. So chromosome 7 has been added here. And you can remove these panels by right-clicking up there and say remove panel. Okay, so now we're down to 2. And now we're down to 1. All right, so that's the end of how you explore true structural variants. I just want to show one more example, which is a false structural variant or an artifact that was identified by our secondary analysis software. So again, I'm going to start by going to the region navigator and putting in the breakpoints identified by our software. All right, so red bar on top, going to zoom out so the entire thing is in view. And I'm going to group by pair orientation. Well, nothing happened, and that's because none of these reads in this region are oriented improperly, meaning all of the paired reads face each other. Now you can see that the reads from this insert are red, indicating that it's a larger insert size than expected, suggesting that there might be a deletion here, but there's only one of those. All right, so right off the bat, I'm going to show you what a genomic region looks like when it does not have a structural variant. There's no soft clip bases here. So that tells you that there's a problem with this call. There's one. Okay, so we can blat those. It's too short, so we're going to have to copy left clip sequence and use our motif tool, but that doesn't always work, so let's just see what happens. All right, nothing there. There's going to be soft clip bases from time to time, even in genomic regions with no structural variant, because again, this is blue. Blue means that it's a smaller inferred insert size than expected. Either it actually is a very small insert, or there's a large insertion here, and there are soft clip bases here, so we could look at that. But like I said, there are even genomic regions with no structural variant will sometimes have soft clip bases because some sequences don't align well. They're poor quality or something like that. Let's just try copy right clip sequence, find motif tool. Let's just put it here and back out. Yeah, again, it didn't really give us any useful information. So I'm going to delete these because they're not providing us with anything. So this all looks pretty normal. You know, there's a couple anomalies, but not like in the last couple examples with true structural variants. The only way you could have a structural variant with no soft clip bases is if the insertions, inversions, translocations began exactly at the start of an amplicon and ended exactly at the end of an amplicon, so that it was for every single amplicon, so that the entire amplicon could be aligned to the reference genome. Now, maybe it's being aligned in the wrong place, but we wouldn't be able to tell if the amplicon were the exact size of the structural variant. But see, all of these amplicons, all of these inserts are different sizes, so there's no way that could happen, right? Because even if that were true for one of these, it wouldn't be true for the rest. There would still be a part of the insert that spanned the breakpoint and so was poorly aligned. So that in and of itself is enough to demonstrate that this is not a structural variant. All right, so that's my overview of how to view and interpret structural variants in IGV, and that concludes the IGV tutorial.